I can see your screen.
Okay, good. Thank you very much, Abik, for the kind introduction. Thank you, Lech, also for giving me the opportunity to present here. Before I start with my actual presentation, I would like to answer your question or your statement about software hacking of images, Lech. In fact, it is a very good point. And there has been an initiative called the OME, the Open Microscopy Environment, where vendors come together and work on a joint format so that there is there's an easy exchange between data and images possible between the different platforms. So I just wanted to bring this back to memory and also state that my company, TissueGnostics, has just 2 weeks ago released the 7th generation of our software. And this supports now, fully supports what is called the OME-TIFF file format. So that makes a mutual exchange between the solutions and platforms of different vendors much more easy, and there is no need to hack the file format. But let me now start with my presentation, dealing with the topic of quantitative tissue cytometry. There are indeed many, many applications, and I will try to give you a short overview of what we do and then focus on applications of immune phenotyping and use of machine learning technologies for recognition of tissue metastructures, which is generally termed also histomorphometry. Although there have been several talks in this seminar, in this conference, especially this morning, with reference to TissueGnostics, I would still like to give a very brief introduction. I'm one of the co-inventors and co-founders of TissueGnostics. Our corporate headquarters is located in the city of Vienna in Austria, in this nice building. We have international branch offices. In Romania is mainly software development, at the US East Coast and West Coast, as well as in Asia Pacific, our sales representatives or sales offices. The company has in fact been founded in 2003, so we celebrated our 17th anniversary this year. And as of today, we have installations in 31 countries on all continents except Antarctica. As of last month, we had more than 1,500 reference papers published from research groups from 58 countries. You can also find them on our website in a searchable database. And I would like to emphasize that TissueGnostics is not just a software company. We are in fact a manufacturer of medical devices and have been certified for ISO 13485 first time in 2008. And this is the main quality norm for medical instruments. And in the previous presentation, there has been an extensive review of this slide scanner competition. And in fact, I would like to mention in 2016, in the discipline of image analysis, TissueGnostics won the first place in this international scanner contest from Charité. Now, what is TissueFAXS cytometry? I may assume that all of us are aware of flow cytometry, which is a technology that has been available for decades. If you go to PubMed or any of these literature databases and search for FACS, F-A-C-S, or flow cytometry, you will literally find hundreds of thousands of publications that have used this method of research in their studies, which are described. The main advantage of flow cytometry is that it quantifies staining per cell, which permits a phenotypic and functional analysis. With one major limitation, the cells have to be in suspension. So if we look at a human body, there is only one liquid tissue. This is blood, or maybe you call also lymph as a tissue. But the majority of our tissues, and I assume that our pathologists will agree, is solid. And therefore, you cannot properly analyze solid tissues, be it skin or liver or lung or kidney or bone or whatever, by flow cytometry. There is now another method called image cytometry, which offers the same type of analysis, but this time the cells are in tissue sections, and the tissue sections are on slides, and the slides are scanned. So we have heard a lot about slide scanning in this conference, especially in the last presentation. And what we do is we digitize or use the virtual slides, which come from slide scanners, and analyze the images. And this is called image cytometry. Now, TissueFAXS Cytometry, that is in fact image cytometry, the specific way TissueGnostics is doing it. I would also not like to miss the opportunity to point to something which might be seen as critical. In 2016, a survey was published in Nature among 1,500 scientists, indicating that 70% of the researchers have problems in reproducing published data. And another 20% even have problems in reproducing their own data. So this is what is generally referred to as the reproducibility crisis. And the authors of this survey call for a better mentorship, which is the duty of universities to teach properly how to do research, the proper use of statistics, which is the responsibility of each PI, each lab, to do this and use statistics in a proper way, but also a call for more robust experimental designs, including instruments. And this is in fact where TissueGnostics comes into play with our TissueFAXS platform. TissueFAXS is not a single instrument, it's rather a family of products in different configurations, scanners and analysis for bright field only, for fluorescence only, or the combination of both. It's called TissueFAXS Plus. As you can see, the hardware core of our TissueFAXS is a Zeiss microscope. In fact, TissueGnostics is OEM partner of Carl Zeiss Germany. So we offer the optical quality of Zeiss microscopes with the ease of use of a slide scanner with a high degree of automation and reproducibility. This is one version based on an upright microscope, which can handle up to 8 slides, 6 different objective lenses, including oil immersion objective lenses. We also have an automated oiling system. And like with any slide scanner, the immediate result of the scanning process, but this for us is only the intermediary result, not the final result, is a virtual slide. As we can see here, an entire mouse leg. And like with Google Earth, you can zoom through it, navigate through it, zoom in, zoom out. This is known and it's standard in every slide scanner. In TissueFAXS, there is also an inverted version, I like inverted, where you can also use slides by putting the slide upside down or work with micro well plates. So in case if you have adherent cell cultures or even want to do live cell imaging. We also have a high throughput version of our instrument with a slide loader for 120 slides. And each fluorescence configuration of TissueFAXS can be equipped with a spinning disc confocal for doing whole slide confocal imaging. Here we see an example of an entire mouse brain. And if you look now on this region here, this is the so-called third ventricle. When we zoom in, then you see the actual quality of what has been acquired. And this would also be a set stack where you can navigate through different focal planes within that tissue. But what makes TissueGnostics is obviously not the Zeiss microscope. It is our software which automates the scanning and offers the image analysis, or more specifically, the image cytometry. And I would like to give you an overview of what is possible. And let's start with the, let's say, easiest application of quantifying a molecular marker that is expressed in the nucleus of a cell. For this, we use the nucleus in bright field, typically stained by hematoxylin, in fluorescence, typically stained by DAPI, Höchst, or markers like TREG5. We use the nucleus as a master channel to identify the individual cell and then quantify how much staining is associated with it. I invite you to look at this image, and in this virtual space, we cannot really do it, but I invite you to look at it and guess how many cells would you say in percent are positive for the brown marker, which by the way is Ki67, so a proliferation marker, and it's obviously column. So how many cells are proliferating? How many of the blue nuclei are stained in brown? And we made a survey among more than 100 scientists, and believe it or not, the answers that we received ranged from 1% to 40%. So some people think 1% of the cells are proliferating, others think 40% of the cells are proliferating, which I would say is quite a significant difference. And this points to the main challenge associated with visual evaluation. And this is that you always get a range of estimations rather than a reproducible measurement. And this is exactly where TissueGnostics comes in. By using, as I said, the nuclear marker as a master channel, the yellow contours show you here what the software has identified as single nucleus. And then we quantify how much blue and brown is present. It also works for other chromogens. In this case, it's the well-known DAB with the peroxidase staining system, so the color is brown. The algorithm also works for other colors like green or red or purple. And the measurement is shown now in something we would call a FAXS plot, a dot plot from flow cytometry, but it is each dot that you see here represents a single cell in that column tissue. On the x-axis, we see the mean intensity of the blue marker, the hematoxylin. On the y-axis, the mean intensity of the brown marker, Ki67. Here is this famous line known from flow cytometry called the cutoff. That has to be set in reference to an isotype-matched negative control, and it determines the specificity of our staining. So all cells with a mean brown intensity below 20 will be considered negative. And this is the population of the Ki67 positive cells, so those which have a specific brown staining. And you see the amount of positive cells is 14.5%. And the important point is, you will always get the same result. Whether you do the analysis on Monday morning, on Wednesday noon, or on Friday afternoon, if you do it here in Vienna, in Nottingham, in Sydney, you will get 14.5% rather than an estimation anywhere between 1 and 40. Now, this was an example of a nuclear expression. What if our molecule is expressed either also or only in the cytoplasm of a cell? Then obviously it makes no sense to measure the nuclear area, because then we miss the actual expression of the marker, which we see here in an example of a skin cancer research project. We see on the left-hand side the normal skin. The marker, by the way, is called COP1, but that's not important. Important is the methodology. And we see the marker is constitutively expressed on the basal layer of the epidermis in normal skin. After UV irradiation, so after sunbathing, the expression of the marker increases in two ways. First, the amount per cell increases, so the staining becomes brighter, but also more cells become positive. Now, normally people would look at this and say, this is 1+ positive and this is 2+ positive. And then maybe your colleague comes and says, nah, I would say this is 3+ positive. So rather than discussing how many pluses you assign to a sample, TissueFAXS Cytometry will provide you an observer-independent measurement. On the left-hand side, we see the measurement result of normal skin, so of this sample. And on the right-hand side, we see after UV irradiation. And what we see at a glance are two things. First, the number of positive cell increases from 16.78 to 44.73%. So this is what we visually perceive as more cells being positive here. And what we also see is that the maximum staining intensity, which is in around 90 in normal skin, shifts towards higher values around 130 after UV irradiation. So this positive population increases also, as we see here in what is called the quadrant statistic, by its mean intensity, by the percentage of positive cells, by the total count of positive cells, 759 versus 195, and also intensity. This means cells per square millimeter of tissue. Now, this is done by using the nucleus as a master channel again, but then creating a secondary measurement mask around the nucleus in order to capture the cytoplasmic signal of that staining. One of the features that our clients around the world appreciate most is the ability to track back and forth individual cells between the virtual slide and data space. We see here a renal allograft rejection. In green lymphocytes, in red are monocytes. And if we click, the contours here indicate what the software has recognized as individual cell. And when we click on a cell, that starts blinking in the scattergram. So we see here lymphocyte positive, monocyte negative. If we click on a red cell, monocyte, we see lymphocyte negative, monocyte positive. Following our motto, from image to data, this is a step forward, and thus we call it the forward connection. It also works the other way around. From data space back to the image, we can make a gate, like in flow cytometry. This is a selection of a subpopulation of cells, and this will then be highlighted in red in this case, and we see where a certain subpopulation is located in the tissue. So there is a link between the phenotype and the location in the tissue, the histology. This is the backward connection or reverse gating. There are many applications of tissue cytometry, but maybe one of the most important ones is the ability to determine the immune status in situ. With flow cytometry, we can determine the immune status systemically by measuring how many immune cells are circulating in blood. Here we can do this for a specific organ, for a specific tissue. It's also referred to as the phenotypic characterization of tissue infiltrating leukocytes. In this example, we see a 4-color staining of colon, DAPI, the nuclear marker in blue, T helper cells in yellow, cytotoxic T cells in red, and a marker called FOXP3 in green. You may know that cells which are CD4 FOXP3 double positive are referred to as the regulatory T cells or TREGS. So we use the DAPI as a master channel. The green contours show you what the software has recognized as individual cell. And then we measure for each color channel how much staining intensity is present. And this will give us here the CD4 positive cells, the yellow ones. So there are some which are very strong yellow, so they will be up here in the scattergram. Others have only a weak yellow staining. They will be further down, just close to the cutoff. And all those which do not exhibit a specific CD4 staining, so shown here by the green contours, will be in this population. So this is the CD4 negative. The same can be done for CD8 cytotoxic T cells, and ultimately for the CD4 FOXP3 double positive cells, in order to determine co-expressions. And obviously this can be done with any marker combination. This is just one example which is relevant for the immune phenotyping. But the determination of co-expressions, molecular co-expressions on the single cell level, is inherent to the TissueFAXS technology. A very attractive combination, I mentioned at the beginning that we also have a TissueFAXS confocal with a spinning disc confocal for confocal whole slide imaging. A very attractive combination is our confocal, TissueFAXS confocal, with something that is called expansion microscopy. I'm not sure if you have heard about it. That's a technology that has been developed by a group around Ed Boyden in Boston. It has been published in a number of high-ranking journals like Science. And the idea also is called expansion microscopy. It's actually not microscopy. It's a chemical process of a polysaccharide. You have your tissue section stained with antibodies providing fluorescent markers on slide. Then you incubate this polysaccharide structure. This captures the fluorochromes from your staining. Then the tissue is removed, but the staining pattern remains because this polysaccharide structure captures your fluorochromes. So although the actual tissue is gone, the staining is still here. And then you expand, this polysaccharide structure forms a scaffold, and you expand the scaffold. It's like when you blow up a balloon. 2 points close to each other get further apart. So you in fact increase the optical resolution. Now, resolution in optics is determined as the ability to resolve two dots close to each other and see them as two rather than as one. When you blow up the balloon and the two dots get further apart, that means you increase the resolution by leaving the optical magnification and the numerical aperture of your optical light path the same. And therefore, we can expand tissue, like we see here an example of a prostate tissue sample, expanded 4 times, scanned with a 40x water immersion lens and our confocal instrument. So the effective magnification is 160x. We all know there is no 160x lens. Not even the best optical companies can make it due to the laws of optics. But by combining the magnification, and in fact, it's the numerical aperture which determines the resolution, with this expansion microscopy, we can go beyond. And I would like to show you this as an example. You see, this is, as I said, prostate. We see here one prostatic gland. This in green, these are the secretory epithelial cells. This is the lumen of that gland. Here is the interstitium of the prostate tissue. And this is just 25% of the original resolution. Now we go to 50%, and you see the brilliance of the optical quality. We can go even further. Now this is 100%, which means 1 pixel in the image corresponds now to 1 pixel on the camera chip. And what we see here, please note these white dots. Looks like dirt or background, but it's not. These are secretory vesicles which come from the secretory epithelial cells and get secreted into the lumen of the prostate. So we can here literally observe individual secretory events and check the structure of the nuclei, and it's amazing, right? This combination of expansion microscopy with confocal scanning. And therefore, we call this expansion-based super resolution. No, but there is super resolution microscopy, which requires separate equipment, is very expensive, is time consuming and limited to small areas. In the combination, excuse me, in the combination of our confocal whole slide imaging with this expansion microscopy, we can scan entire slides, entire tissue sections in multiple channels at super resolution level. And that's why we call it expansion-based super resolution. With the advent of precision medicine, it has become more and more important to determine an increasing number of markers on the same cell population in parallel. What has been done for decades is that you make consecutive section and stain them with different markers, and then you combine how much of the first marker is present in a certain region, like in a tumor area, how much of the second, of the third, and the fourth, and so on. That's okay. You can determine the co-expression in regions, but you cannot determine the co-expression in single cells, because as you go along the tissue block and you make consecutive sections, you cut different cells. So when you look on tissue section 3, 4, or 5, not a single cell which is present in the first section will be still present because you just cut them away. Therefore, techniques have become increasingly important, which allow to determine the expression of molecular markers of an increasing number, more and more molecular markers on the same cell population. One of those techniques is multiplexing. This is a publication from Yale University, Kim Blenman, from last year in Cytometry, where she introduces a protocol based on our TissueFAXS platform and the StrataQuest analysis software. The basic idea of multiplexing is you take a tissue section, you stain it with 3, 4 markers with a given fluorochrome combination with your standard fluorescence microscope, or in our case, our standard TissueFAXS. Then remove the staining. There are different protocols. One of those is described in this paper. And stain the same tissue again with the same fluorochrome combination, but in the second staining round, each fluorochrome represents a different marker. And then you repeat this several times. So you get multiple markers, maybe 10, 15, 20 markers stained on the same tissue or not on the same cells. So it's really a co-expression analysis. After registering, Alain Pitiu yesterday has talked about image registration. After registering the sample, in this case, it's pretty easy because it's really the same sample, just rescanned. And so you increase the number of channels or markers which are present on the same sample. And what you get, you see here, different marker combinations shown. Looks like one of these famous multicolor flow cytometry samples, but it's not flow cytometry, it's tissue cytometry. Again, each dot that you see here comes from a tissue section. The second technology which can be used to increase the number of markers in the same sample, on the same cells, cell population, is multispectral imaging and spectral unmixing. For this, we use a liquid crystal tunable filter that acquires a lambda stack. You are probably familiar with the term set stack from confocals. A set stack is when you acquire the same region, the same field of view at different focal positions. A lambda stack is when you acquire the same region at the same focal position, but in different spectral intervals. Typically a step width of 10 nanometer. And then you can mathematically analyze how much of the total emission comes from fluorochrome 1, from 2, 3, 4, 5, 6, 7, 8, maybe up to 10 colors. Here we see the raw data set before unmixing, and after unmixing, it's this. It's actually a complete bile from mouse. And we see here the different T cell zones, B cell zones, monocytes, smooth muscle cells, and also endothelial cells in this cyan color. As a very current publication, or for current reasons, I would like to point your attention to this recent publication from August this year. In Cell, you know, one of the highest impact factor, whatever, 38.6. This comes from the Reagan Institute in Boston, which is associated with MGH, Massachusetts General Hospital, MIT, and Harvard. And they used our TissueFAXS system in the multispectral analysis in order to quantify the immune response against SARS-CoV-2 in COVID-19 patients in the lung tissue. And I don't have the time to go into details now because this presentation alone would be an hour probably. But what I want to show you is, actually it has been in August, it was published online, and yesterday it was really published in Cell. So this is really top recent. Although the outcome, I have to say, is not very encouraging because it shows that the concept which many countries currently follow, the concept of herd immunity, will probably fail in COVID-19, as they are showing here that the SARS-CoV-2 virus evades the long-term immunity, you know, by avoiding the formation of follicles with germinal centers. As I said, I don't have the time to go into details. If you want to know more, please refer to this publication published yesterday. But this is one example that shows how TissueFAXS cytometry can support or can perform immunophenotypization of tissue-infiltrating leukocytes. In the latest version 7, which I mentioned at the very beginning, that also supports the OME, open microscopy environment image types, we also have an extended machine learning module and a new algorithm for nuclear segmentation, especially in such lymphatic tissues, which tend to be very, very dense, very high cell density. Sorry, I apologize. I have some asthmatic problems since 30 years, but when you cough during Corona, it's always a problem. So this new algorithm is based on deep learning. We see here an example of lymphatic tissue. When we zoom in, we see here the high density of single cells, of lymphatic cells. This is the nuclear marker only. And here we see that the new deep learning nuclear segmentation algorithm gives an unprecedented accuracy in finding single cells, even in areas with high concentration, high cellular density. Our new machine learning algorithm is able to classify tissue, like here, skin, and multiple classes are possible. Not only one or two, but in this case, we have 7 different classes indicated also by the different colors. A simple training, this is epidermis, this is dermis, this is subcutaneous fat, or whatever you're looking for is enough. And within minutes, the software is able to classify your tissue sample. And finally, I would like to show you how with our software you can analyze histological structures in combination with the single cells. Now looking here on the tumor microenvironment and the spatial phenotyping, as it's called, we see here a colorectal cancer stained with 5 markers. Dark blue is DAPI, light blue is cytokeratin, so this is the tumor area in this case. In red are the CD4 positive T helper cells, in green FOXP3, and occasional white events are Ki67. We identify the individual nuclei, as I have shown in the beginning, by using the nuclear marker as master channel. Then the tumor area here in orange, so we have different, what is called measure masks, for single cells, for nuclear area, also for the tumor stroma, and then can ask the all kinds of questions. This is from a client in at University of Western Australia in Perth, and they were analyzing T helper cells and regulatory T cells, which role they play in this colorectal cancer. And here we see an output, a result, as a readout. All dots that you see here are CD4 positive T lymphocytes. So this is what we call the input gate. On the x-axis is the size of the nuclei. On the y-axis, the distance of each nucleus from the next tumor area in micrometer. And that allows us to generate here gates. Like this gate will contain all those CD4 positive cells, which are 1 to 25 micrometer away, 25 to 50, 50 to 100, and so on. And here you see the output. In total, there are 7,234 cells, CD4 positive cells in the tissue. 2,267 are in a range up to 25 micrometer around the next tumor area. 1,593 or 22%, 25 to 50, and so on. So here is in total number, here is in percentage, here is in density per square millimeter of tissue analyzed. And we see how the percentage decreases so above 100 micrometer. That shows the vast majority of CD4 positive cells is really in the vicinity of the tumor areas. And this we see here visualized in these images. In orange is the tumor area, and in red are those CD4 positive lymphocytes which are just outside the tumor, next to the tumor, but not further away than 25 micrometer. Now we see those 25 to 50 micrometer away, 50 to 100, and so on. So we can really do a very detailed and differential analysis on whole tissue samples on the single cell level. This is another way of showing it, the distance zones or proximity areas, as they're called, from the tumor. Here you see the distance, green, 0 to 25, 25 to 50, and so on. And this then can, of course, be plotted in terms of, this would be the cell number in the tumor microenvironment, how many cells are in which distance from the tumor. I mentioned this spectral imaging approach. Here we see an example of a 7-channel spatial phenotyping, the very well-known immunological protocol for the phenotyping of the immune response. This would be the original image, which is, you see, has a lot of overlay. After this spectral unmixing, we get a beautiful, clearly separated, specifically stained sample, We can now look on the individual markers that have been stained here. I don't have the time to go into details, but again, based on this basic strategy to use the nucleus as a master channel, we get a FAXS-like analysis for all cells, CD4 population, CD68, PD-L1, and so on, for all cells in the tissue section. And again, you can click on any cell and it will start blinking. So you know exactly where this cell is located. These are, again, the proximity areas, and these show then the distribution. Over here, we see the spatial distribution of the cells in your tissue sample. Here we see the population of CD68 positive macrophages, and in red is always the content of this gate using this reverse gating feature. Yeah? So the cells with red contour shown here come from the active gate. Yeah? Here CD68, CD8, cytotoxic T lymphocytes, CD4, T helper cells, CD4, PD-1 double positive cells. And here you see that in the first gate, which shows PD-L1 area, so the size of the cells versus PD-L1 mean intensity. This gate contains the PD-L1 positive cells. This gate is defined as an input gate in the second scattergram. That means all cells we see, all dots, and each dot represents a single cell, all dots we see here come from this gate. So all dots here are PD-L1 positive cells. Now we take this, what is called the lower left quadrant from our 4 quadrants, upper left, upper right, lower left, lower right. We take the lower left quadrant as input gate for our third scattergram, which shows here the CD68 and CD8. These are the double negative cells, CD8 negative, CD68 negative. And if we make now the backward connection on this gate, we see now in red contour those cells which are PD-L1 positive, CD4, PD-1 negative, and CD8, CD68 negative. So this is what is also called gate arithmetic, positive in one gate, negative in other gates. And so you can visualize where a certain subpopulation of cells is located in your sample. This can also be shown with such lines. So in blue, this is the proximity analysis. In blue, we have the tumor cells. In this whatever pink color are the cytotoxic T cells, and the lines indicate those tumor cells which have cytotoxic T cells within the 30 micrometer margin or vicinity. Here we see the same tumor cells in blue, cytotoxic T cells in pink, effector T cells in this kind of yellow or green, and the dark green are the regulatory T cells. Or here we have effector T cells in this yellowish color, cytotoxic T cells in green, and regulatory T cells within 30 micrometer of effector T cells. So a very detailed and differential analysis, including phenotypes, subpopulations, and spatial orientation. So where in the tissue are certain cells or is a certain cell population? And from this, we can calculate what is called an aggregation index. That is the ratio of the connecting lines per the number of cells which express a certain marker. To conclude my presentation, I may point out that our method has been published. I mentioned at the beginning more than 1,500 papers, but among those are also some papers which focus on the feasibility or the proof of principle, let's say. The first one was published by myself in 2006 already. The technology was, the instrument is called TissueFAXS, but the technology is called microscope-based multicolor tissue cytometry, MMTC. We used here peripheral blood leukocytes stained for these markers and analyzed them in a standard Becton-Dickinson flow cytometer versus our method. And you see the results obtained by both instruments show a very good correlation coefficient. Also, the client from Perth have analyzed or manually counted the FOXP3 positive cells, in their samples. 3 human observers have counted, and as you see, although human evaluation is still considered the golden standard, it comes with a problem, because when you have 3 people count, you get 3 different results. So the median was calculated from these 3 observers, and then the median compared to the result provided by the software. And this shows also very good correlation. Okay, very good correlation efficient. I would like to acknowledge and thank our academic collaboration partners around the world. In particular, Professor Mohamed Elias, with whom I have, TissueGnostics has been collaborating for years in the Marie Curie project 8th Pass on digital pathology. But we have also been, or still are, members in a number of other Marie Curie projects dealing with the calcium sensing receptor, with translational oncology. This was headed by a group in Cambridge, this Medical University of Vienna, this Technical University in Aachen, INSEM, the European Network on Cell Migration, and currently headed by Trinity College Dublin, a project dealing with big data analysis for clinical benefit by machine learning. And finally, I would like to thank the European Union for providing the funds for this fascinating European multinational research projects. Thank you very much for your attention, and I'll be happy to take any questions.
Thanks, Rupert, for